Hi, my name's Adam Jussila, and I'm a field bioinformatics scientist at Singular Genomics. Today, I'm going to be telling you about designing and analyzing high-fidelity spatial experiments on the G4X. This presentation will cover G4X curated panels and panel customization options, as well as G4X tools for exploration and data manipulation, and finally, G4X outputs and integration with common single-cell analysis tools. Our G4X custom panels come in two flavors. We have pre-made, curated, in-house validated panels covering a variety of immuno-oncology contexts in human tissues, and more recently, a new pan-mouse panel. Each of these panels can be customized to a small degree, allowing you to add on specific genes that cover biology of interest for you. Additionally, though, if these panels do not appeal to you, we allow full customization of up to 500 genes. These curated panels focus on kidney, lung, colon, breast, pancreas, and skin. Each of these panels is built on a base of around 50 stromal cell genes and 150 immune cell genes. The plexity of them ranges from about 290 to a little over 400. And each of these panels is designed using our own in-house experts to do literature curation and data searching to find the optimal genes to cover each of the core phenotypes and cell types present in these cancers. If these panels themselves are something that appeal to you, but are lacking in a few key genes for your research, we encourage you to look at adding up to 50 user-selected genes to any one of these panels. If these panels, however, do not cover the specific disease or tissue context that you care about, we allow a custom module to be built of up to 150 genes built on top of our immune microenvironment panel covering these 150 and 50 genes that I mentioned earlier. Additionally, though, we do offer the full customization option that I'll talk about in a few minutes. These curated panels are extremely high quality, having been tested extensively both in-house and at our early access sites. I want to tell you about one of them just to give you a sense of the kind of quality you can expect from your own panels if you were to work with ours, pre-made ones like this. The human pancreas panel is around 400 genes. It covers 10 core cell types spread across four pancreatic cell types like alpha, beta, gamma, and delta cells. We use literature curation to identify key cell types and key genes associated with those cell types across both normal pancreas and multiple cancer types. We have numerous marker genes for each cell type, ensuring a robust detection of each of these unique cell types within your tissue. Shown on the bottom here is a core transcript or core statistics table covering three different samples that we ran on our home systems using this panel. Among these, we have normal pancreas, PDAC or pancreatic ductal adenocarcinoma, a common cancer type, as well as PNET or pancreatic neuroendocrine tumor, another common cancer type. Each of these samples got hundreds of thousands of cells per tissue block, tens of millions of transcripts, and really robust median transcripts per cell over 100 transcripts per cell. The median unique genes per cell is a little bit lower, you might notice, for the normal pancreas. This is primarily because our panel is designed to target cancer cell types in particular, and so our normal pancreas will not likely have many of these transcripts that are unique to the cancers that we're observing. As such, this is an expected result. For the PDAC and PNET tumors, we see a significant uptake, around 50 unique median transcripts per cell. This is quite good for our assay and something that we're really happy with. Overall, pancreas is generally a high-performing tissue for us. Additionally, we have our false discovery rates for NCP or negative control probes, as well as our false discovery rates for NCS or negative control sequences. Negative control probes essentially tell you the off-target binding frequency, and negative control sequences are used to kind of measure a background of misidentification rate. We're consistently getting FDRs below 0.5, which is something we're quite happy with. Now, moving on from this sort of statistical perspective here, I want to show you the actual pictures. First, here we have our normal pancreas, where we see clear and distinct morphology consistent with a healthy and normal pancreas. You can see clear spatial features mapped across this tissue block, which are consistent with our expectations. We're also very pleased to see that in both PDAC and PNET, we can easily distinguish the tumor populations. In particular, this PNET sample, which is sort of a solid tumor, shows this bright blue population of these neuroendocrine tumor cells. In the PDAC, we have a slightly less easy to detect or visually obvious phenotype, where we have these PDAC epithelial cells woven throughout this tissue. You can see here that in both instances, our G4X assay is able to easily distinguish cancer from normal. Furthermore, when we look at our differential expression heat map here, we can see a sharp diagonal line indicating that we have high specificity of our marker genes to the intended cell types. Overall, samples which look like this are very likely to come from any one of our curated panels. We've tested them extensively, as I mentioned, and I hope that you'll consider using them if you have specific biology that's covered by the pre-made panels that we offer. Now, if you're still interested, though, in pursuing full customization, we have two different levels. The base level we call Craft Custom Panel Process, which allows you to design for up to 500 genes. You submit a gene list to us. We process them, generating a list of candidate sort of padlock probes to be used on our G4X assay. And we guide you through this design process, giving you detailed reports on the target gene performance. These reports allow you to review what we've done and come back to us with critiques or changes you'd like made. And we do this in an iterative fashion, allowing us to sort of hone in on exactly the panel that you want to come out of this whole process. When you're happy and you're ready to move on, you have us move to order the oligos, and we ship them directly to you. This whole turnaround time on this process is around 4 weeks from submission of your gene list to order of the oligos. The signature custom panel process builds on the craft process with these same three core steps, except at the end of this process, we add one final key step where we validate each of these probes in-house. So this in-house validation ensures an extremely high quality panel. It's sort of like a high purity panel where we've removed any potential errors or off targets by actually testing this process on real tissue in-house. The signature process includes custom protein add-ons, again, with in-house antibody validation. Because of these validation steps, though, the turnaround time is between 8 and 10 weeks. Now, I want to tell you a little bit more about a couple of key steps in this process. The first of these is the PLP design and sort of customer report, and the second of these is the in-house validation, which I'll get to in a couple of minutes. Our panel designer works in the following manner. After you submit a gene list to us, we design candidates for each of these gene targets, ending up with tens of thousands of possible candidates. These cover all possible sequences across each of these genes with little or no filtering on the first pass. We then perform core filtering, identifying sequences which will perform well in our specific G4X chemistry, allowing us to filter for things like GC content, melting temperature, and so on. Once we've narrowed this down a little bit, we then do in silico off-target binding analysis, selecting for the highest specificity probes for each gene target. After this, we take this much narrowed pool of probes and we sort of combinatorially mix and match them to find the best combination that have the fewest kind of collisions and off targets with each other to give you the best whole cohesive pool of probes at the end. These core two steps are where we report to you and get feedback from you on exactly how you want us to proceed for every single gene that's not designing exactly how we hope. Now, if you've chosen to do the signature process, we have one final step that I mentioned, which is that experimental validation, where we run on real tissue samples looking for specific spatial localizations of your probes to the regions of interest. As we move through this process, the number of probes dramatically refines from hundreds of thousands at the beginning in some instances down to only a few thousand. This custom probe design report helps us go through this process with you, making informed decisions about exactly what you want to guarantee the highest quality product. Shown here is an example of our custom probe design report. This is for our mouse atlas, or often called our pan mouse panel. At the top, we get some high-level statistics, including the targeted species, the number of probes, the number of genes, the total number of probes, the number of probes minus the number of control probes, and the number of rounds. Some of these information will stay consistent across your panel designs, but the sort of core of this here is going to be variable based on what you're designing for. As we scroll down, we get some summary metrics telling us about the overall high-level performance of your panel. In particular, we highlight the number of genes, which we have no coverage and low coverage for. In general, the vast majority will have standard coverage, which to us or which to you should be interpreted as these genes were designed for within specifications, and we have no concerns about our ability to detect them. It's not uncommon, however, to get at least one or two no coverage genes for the larger panels. This report then allows you to go in and dive into each one of these low and no coverage genes to identify and understand exactly what the performance is and exactly what you might want to change or not as you move forward with the panel. I want to emphasize, though, that these low coverage genes do not mean that these genes won't be detected or that we'll have difficulty detecting them in our assay. It's just our way of letting you know that if these are critical genes to your core biological questions, then you might want to consider iterating on this and changing some parameters or making some allowances for off targets that will allow you to guarantee detection of your key genes. Now, for each of these low and no coverage genes, you're going to get these little sort of interactable panels popping up here in yellow and in red if they're no coverage. We can click on this dropdown and look through these genes. Now, let's just for a moment imagine that one of the genes that you care the most about is CHIL3. It's really key to some biological question you're asking. You see this in the warning or low coverage area, and you want to understand a little bit more exactly what's happening. So if we click on it and look through, we're going to get a gene level probe statistics report for this one specific gene. And not actually just this one gene, but every single gene. You see here, it starts by telling us the number of probes, some transcript isoforms, exons, and exon-exon junctions covered, and then gives us a sort of staged nested pie chart that tells us at exactly which stage we lost what percentage of our original candidate probes. You can see then how we ended up with our final 1% of passed probes. Now, if we look at this and we decide that three probes is really just not sufficient or is maybe concerning for us for a key gene like CHIL3 that we care about, we then encourage you to come down here and check on these off-target hits. Here we list the off-target hits ordered by highest number of off-targets down to the lowest number of off-targets. So here 733 means that we have 733 of these total probe candidates colliding with the CHIL4 gene. Now, let's just again imagine that CHIL4 here is actually a gene that we're very familiar with, right? It's in the same family as CHIL3, and maybe it's a lesser expressed isoform or even something that in our specific tissue context is not expressed at all. So we're actually okay with accepting off targets here. This is one of our sort of tunable knobs that we can work with to enable you to change something about the panel design to fit your biology better. So then you can report back to us and say, I'm okay with accepting an ambiguous probe that's either CHIL3 or 4 when we run this analysis, allowing me to more robustly detect the core genes that I care about. We'll then rerun it and iterate, hopefully getting more probes and making this jump out of that kind of low coverage group here. Now, there are more tunable dials here, but we can work through that and talk to you guys about that if you decide to move forward with this process. We also allow you to look through every single gene, not just our low coverage and no coverage genes. So for example, if you wanted to look at something like CCL3 here, we can click on this and get the same level of detail from our probe report, detailing exactly how we got to our final set of candidates. Again, allowing you to look at things like off targets down at the bottom here. I hope this gives you a sense of the level of information we try to provide for you during this custom panel design process. These reports are generated at each step, and every time we make a change, a new report will be generated and sent to you. The whole goal here is to allow you to make the most informed decision to make sure that your probe panel is as customized and as specific to your sort of interests and your research as possible. To give you a sense of how good the quality is coming out of this process, I want to tell you about our recent and soon to be released pan mouse panel. The pan mouse panel was designed explicitly using the craft panel design process with no in-house validation. This you can think of this as sort of a proof of principle for this whole sort of custom design process. We identify genes from this much in the same way we expect you will be identifying genes, through a combination of literature-derived and data-derived gene lists using publicly available atlas-style single-cell and single-nuclei datasets. We're really excited also that this pan mouse panel, our first ever non-human pre-designed panel, has also demonstrated comparable RNA performance to all of our human panels. You see down here another core metrics table across 5 different tissues, which are the 5 tissues that we've designed and targeted for in our pan mouse panel. We have colon, kidney, lung, spleen, and brain. Here we have tens of thousands of cells, again, for each of our tissue blocks, around 10 million transcripts per tissue block, and really impressive median transcripts per cell. You'll notice here that the spleen is a little lower. There's a relatively significant amount of variability between tissues, but 60 transcripts per cell on average is actually still quite respectable and something that we're really happy with. Our median unique genes per cell is once again looking quite good, with between 40 and 50 for all tissue types, and our FDRs are consistently below zero point 0.5, which we're really happy with. Now, just to dig into this a little bit deeper, I don't want to just show you our UMAPs. I would love to show you a little bit more about this. When we take these UMAP embeddings and we subcluster, we see again really robust detection of distinct cell types that we're expecting, which isn't surprising because when we look at our heat map here, we see really nice sort of specificity of our marker genes to our specific cell types. When we look at the actual spatial maps, though, this is where we get the really exciting results. When we look at the colon, for example, we see these really beautiful immune hubs popping out with really distinct and sort of morphologically correct looking features on the colon sample. In the kidney, we see these cortical layers kind of descending inward throughout the kidney. Lung, we can see these what are presumably some sort of vasculature. In brain, we can see this sort of thin, membranous looking kind of areas of tissue that look like they're astrocytes. And spleen, again, we see distinct spatial patterning. And across the board, we're really excited to see how well this is performing. Because remember, this is a pan mouse panel. This is not tissue specific in the sense that it's a panel not just designed for one tissue, but designed to target many tissues. Overall, I hope you can get a sense from this that this is this if this is the result of our process of our custom craft panel process, that this is something that's absolutely worth your time and energy because the results speak for themselves. Now, as I wrap up, I want to say one final thing. I mentioned in our signature custom panel process that we have in-house validation. This takes the form of something that we call spatial correlation analysis. This probe level validation allows us to leverage probe level data and spatial clustering statistics to identify probes with off-target binding. These probes are removed from the panel manually before we release the panel to you in our signature panel process. And while this is something that we do for you for the signature panel process, I want to note that these can be bioinformatically removed manually if you've done the craft panel design. So this does not exclude you from doing this just because you didn't choose our signature process. Here we're showing 12 different probes across a single gene called C1s. You'll see distinct spatial localizations of almost every probe, but you might notice that probes 5 and 9 are distinctly different from the rest. Now, visually, this is obvious, and to us, this indicates that there's likely an issue where these two probes are not having the specificity to our intended target that we hoped. Then we can then go in and perform a statistical analysis using Ripley's case statistics, looking at each pairwise probe combination. When we find outliers in the statistic, this suggests that there may be binding to unintended targets, thereby allowing us to uniquely go in and remove these probes from the panel. Again, this is something you could do bioinformatically, though it's an extra and added step that you'll have to do in your own analysis. Now, this is sort of the core things that I wanted to tell you about our custom panels, as well as our pre-made and curated panels. And as I move on, I want to sort of jump to the later part of the process. So after you've designed a panel, you've run some samples and generated your own data. I want to tell you a little bit about how we've made tools that'll help you dig into your data faster than ever. This next section is talking about G4X data, tooling, and analysis. The G4X was designed to produce really standardized data formats that are easy to work with and easy to use. In fact, our goal is to produce data formats and data types that you're so familiar with already that it doesn't require you to learn anything new. One of our standard outputs is actually designed for one of our custom in-house tools that we call the G4X viewer. The G4X viewer is a dynamic image viewer that allows you to look through multimodal, multi-layered images, sort of immediately exploring and analyzing the data. It's designed specifically for G4X data and takes in a number of files that we produce by default. This is one of our core tools that we produce for you, designed to help you explore the data. You can visualize fluorescent H&E, multi-layer protein stains, transcript information, and cell segmentation, and more, all in the same place. Additionally, we want to help you immediately jump into QCing your data and determining whether or not the quality is meeting your expectations, or if there are any concerns that you want to address with us or at your own in your own labs. This is found in our summary HTML that I'll talk a little bit about in a moment. The G4X viewer is currently hosted on a public URL on our website, requiring no local download unless you choose to do so. We also offer download from our GitHub, as well as multiple tutorials to walk you through the core functionality of the G4X viewer. This includes a slideshow as well as a 30-minute video tutorial. Some of the core features that I want to highlight here, though not exhaustive, include multi-channel, multimodal visualizations, the ability to customize the colors of any given channel, including protein stains, transcripts, and so on. Dynamic and sort of snappy data exploration. This is designed to be used on any sort of personal computer or HPC of your choice. It should be responsive because of its ability to dynamically load the data at any sort of zoom level and navigate around seamlessly. We also enable screenshot functionality and ROI selection. The ROI selection can be done on UMAP, spatial maps, or flow cytometry data. And the last thing I want to highlight is that we also have many transcript layer manipulations, which include selection of individual transcripts or isolation of transcripts to be visualized on the spatial maps in FH&E. These are just a few features, but I hope you get the sense that our G4X viewer is designed for G4X data and for your ease of use. The other thing I wanted to tell you about is our summary HTML. This summary HTML is optimal for rapid QC of your data. It contains 5 core tabs. The first of these is the Summary tab, which shows summary statistics and interactive FH&E stain visualization. The second tab, Decoding, gives you decoding statistics, transcript counts, and probe quality scores. The third tab, Cell Segmentation, gives you cell segmentation statistics. The fourth tab, Analysis, gives you transcript information, including counts, UMAP, interactive latent clustering plots, and differential gene expression tables by cluster. The final tab is an image QC, which is kind of like a gallery of grayscale protein images across the tissue blocks. Let me give you a sense real quick of what this would look like. So shown here is one of our lung cancer samples that's about to be released as a publicly available dataset. As I mentioned, the Summary tab has core statistics, as well as an interactable FH&E, where we can toggle various options and interact with the plot, drag it around, zoom, and so on. We can change tissue opacity, heat map, color scaling, and so on. But the whole point here is to give you a quick and dirty way to look at your data and get a quick sense of exactly how it looks, how it performed. In the Decoding tab, we start with a rank plot showing you gene level information about counts and the total detection of genes and what rank order it is in the assay. You can search this by typing any gene name and flip through a set of tables until you find the one you care about. This also includes NCP and NCS. We also have probe quality scores, stratified by whether they're a panel probe, negative control probe, or negative control sequence, allowing you to rapidly identify transcripts that might be concerning to you based on their performance relative to the controls. In the cell segmentation tab, we have interactable histograms showing you information about the cell size, transcripts per cell, and genes per cell. This can be used to identify whether the segmentation we're performing is fitting your cell type and your tissue type effectively. In the analysis tab, we have two different pairs of plots showing the transcripts per cell overlaid on spatial map and UMAP, as well as the latent clustering overlaid on the same two plots. The latent clustering has multiple resolutions to allow you to toggle through and see which resolution might fit your specific use case a little bit more appropriately. These are only two pre-made resolutions, and many others have been done in some of our saved out tables. And if you have not found one that you like, you can run your own very easily. Lastly, in this tab, we have differential gene expression by cluster, allowing you to tab through an extensive table detailing which genes are expressed significantly higher in which clusters. The final thing that I want to show you here is our image QC. Image QC allows you to rapidly and easily go through, look at each individual protein stain, and determine whether or not its spatial localization matches your expectations. This is your quick way to identify if there might be any problems or concerns with the performance of the multiomics assay. I hope this has given you a sense of some of the resources we're trying to provide for you to enable your early exploration and rapid QC of the data. We want to make it as easy as possible for you to generate this data and then determine its quality and determine the core biology that you care about. As you move forward, assuming you've passed all QC checks and you're happy with the data, we also want to make it as easy as possible for you to port this data into all the tools you've been using for years already. We want to support both developers in Python and R. To do this, we use two core formats that you're probably quite familiar with. We're compatible with single cell experiments, which is core to the bioconductor packages in R. We also use AND data, which is core to this SC-verse or single cell verse set of packages, among others, all through the Python ecosystem and also some niche packages in R. This means that we port very naturally into packages like Seurat, ScanPy, SquidPy, and SCVI tools. But again, the entire SC verse is available to you through our AND data format outputs. Because of things like our AND data, I want to also give you a quick sense of exactly how easy it is to port our data directly into something like ScanPy and plot your spatial coordinates. These integrations are designed to make it as sort of rapid for you as possible. So imagining here that our goal is to simply plot in ScanPy using only a couple of lines, we do the following steps. It's as simple as importing your libraries, reading in our pre-made H5 data in the customer outputs, performing a one-line transform, and immediately we can call scanpy.pl.scatter, one of the core plotting functions in ScanPy, using default parameters and produce this beautiful spatial map showing log total counts of transcripts. This is not the only functionality, but it's just one example that I wanted to use to articulate that we've designed this to be easy and sort of low input for you to get the information that you care most about from our assay. If there's anything that I didn't cover, or if there's anything that you feel that you want a little bit more clarification on, we also have documentation designed to guide you through your early explorations. This documentation is actively being expanded and worked on, and currently shows detailed information about G4X outputs, how to import it into core libraries, some helpful documentation on some of our tooling, like the G4X helpers, the G4X viewer. And we're currently working to expand our G4X tutorials section, where we give a number of helpful guides on exactly how you can begin to work with your data downstream. The last thing I want to say, and the last thing I want to mention, is that if you're excited about this data, but you're not quite ready to start customizing a panel until you've got a better sense of exactly how it looks, we encourage you to download and explore our sample data sets. At this point in time, if you want to access them, please just reach out to us and you can get links to any of our 11 publicly available data sets, which are covering a variety of tissues and disease contexts. These are rich data sets that have been extensively curated, and you can get a really good sense using our viewer as well as the data itself of exactly what you can expect when using G4X. With that, I want to wrap up and say thank you for your attention. I hope you've learned something today, and I hope you've enjoyed learning about the G4X panel customization, pre-designed panels, and the tooling we've designed to facilitate your easy use and adaptation of our product. I hope you also enjoy the rest of the Spatial Symposium. And up next, we're going to be hearing from Dr. Evan Keller about renal cancer metastasis using the G4X. Thank you for your time.